My name is Helena Hysong, and I conduct research at North Carolina State University. It is a great pleasure to present my literary review article at this conference, Quorum Sensing Inhibition by Delftia tsuruhatensis, a model for fighting multi-drug-resistant bacteria. This literary review was published in the Journal of Applied Microbiology. The primary audience of the journal is those interested in high-quality and impact research relating to novel facets of applied microbiology, pertaining to microorganisms in a wide range of subfields, including medicine, biotechnology, and bioremediation. In this presentation, I'm going to delve into why quorum sensing is such an important topic. What are its implications? Then we are going to look at Delftia tsuruhatensis' ability to inhibit quorum sensing in certain microbial species. We are also going to take a look at the mechanisms that D. tsuruhatensis employs to achieve its inhibitory effects, which will lead us into the future directions of this study. Then we're going to finish out with the potential implications of this research and a quick recap of everything we discussed. We all know that bacterial resistance is outpacing the development of new antibiotics. Antibiotics kill susceptible bacteria, selecting for antimicrobial resistant strains. Gaining insight into the quorum sensing mechanisms that bacterial pathogens use to achieve their virulence may provide a new route for fighting these multi-drug resistant bacteria. New studies have identified a bacterial species that can significantly inhibit quorum sensing, suppress biofilm formation, and attenuate virulence in a highly drug-resistant pathogen. What is even more significant is that this bacteria achieves these effects without damaging individual bacterial cells. This characteristic means that this bacteria has a great potential to serve as a model for designing a novel therapeutic that fights bacterial pathogens in a new way. Quorum sensing is the cell-to-cell signaling mechanism that bacterial species use to communicate by sending out small diffusible messenger molecules. This image is the general structure of an acetyl-homoserine lactone, the family of molecules that bacterial species use to achieve quorum sensing. Recent studies have shown that this signaling ability contributes to bacterial ability to form biofilm. The reason that this revelation is so important is that biofilms are responsible for increasing a pathogen's virulence and contributes to their antibiotic resistance. We know that quorum sensing allows bacterial species a selective advantage that makes them more apt to survive in their environment. So it makes sense that during bacterial competition, a competing bacteria may want to inhibit that bacteria's ability to communicate. It was observed that D. tsuruhatensis does just that, inhibits quorum sensing in Pseudomonas aeruginosa during bacterial competition. Delftia tsuruhatensis has been shown to be able to significantly inhibit quorum sensing, suppress biofilm formation, and attenuate virulence in P. aeruginosa. Moreover, D. tsuruhatensis achieves these effects without damaging individual bacteria cells, an important characteristic in avoiding the selection that leads to antibiotic resistance. While the precise mechanism that D. tsuruhatensis uses to achieve these effects is unknown, there is an agreement that the bacteria sends out a small diffusible molecule, or a group of associated molecules, to achieve this effect. However, there's a disagreement surrounding the identity of the molecule or molecules. These images are of the various diffusible molecules that have been identified by different research groups to be the molecule that D. tsuruhatensis sends out to inhibit quorum sensing in P. aeruginosa. There's even fewer knowledge regarding the actual mechanism that the responsible molecule uses to achieve its inhibitory effects. Before we can use Delftia tsuruhatensis as a model for designing a novel therapeutic, there are a few more aspects of this inhibition that we need to understand. We need to definitively identify the small molecule or group of associated molecules responsible for D. tsuruhatensis' ability to inhibit quorum sensing. Moreover, we need to gain a deeper insight into the mechanistic principles that govern this inhibitory mechanism. While D. tsuruhatensis' ability to inhibit quorum sensing has been well demonstrated against P. aeruginosa, further study should be aimed at identifying the potential range of implications that this model may serve for fighting other pathogenic species. And of course, clinical trials will need to be conducted to test the efficacy of this drug, once the responsible molecule and mechanism are identified. What we learned is that quorum sensing allows bacteria to communicate in such a way that contributes to their virulence and antibiotic resistance. We identified a bacterial species, Delftia tsuruhatensis, that can inhibit quorum sensing in certain bacterial species, such as the highly infectious and drug-resistant bacteria Pseudomonas aeruginosa, and that while future research is needed in order to discover the exact mechanism that D. tsuruhatensis uses to achieve its inhibitory effects, this bacterial phenomenon has the potential to serve as a model for designing a novel therapeutic aimed at fighting antibiotic-resistant bacteria. Thank you.